Okay, so today's lecture is maybe in some sense a little bit non-trivial, at least from that standpoint that I suppose you can probably tell me more about whatever I'm going to consider today to me than I can tell to you. Because I guess you should have covered this material during your course in molecular biology, so I guess you'll recognize many things from this lecture already, and maybe you even know some details that I have not included. And the second thing, well, I don't think we'll do that today, but tomorrow on the second part of this lecture, I guess I will be able at last fulfill whatever I have promised to you and tell you a little bit about CRISPR-Cas. But I'm not sure, maybe you have studied that already somewhere. But I remember that I promised you, so I guess we will get to that tomorrow. Good. Okay, here is some sort of schematic view of the processes that we are going to consider. Of course, the problem is that this is a picture for eukaryotes, and I will today more focus on prokaryotes. So, like some bacteria, and the one bacteria that we probably love the most called Escherichia E. coli. So, I'm not sure how to spell Escherichia or whatever, so let's call it just E. coli. But this is some sort of illustration just to get some idea of the processes we are going to talk about, and these are all the processes that are involved in the central dogma. So let's look first at the eukaryotic cell, and you probably should recognize many of these things. So here is a strand of the DNA. We know that to pass genetic information to the next generations, we need somehow to duplicate the DNA. So the process of replication makes a few copies, well, exactly 2 copies of each of the strands of the DNA. And of course, we need somehow make proteins based on the information that is encoded in the DNA. So here we have first the process of, how do you call it, this one? Okay, I'll give you a hint. Transcription or translation? Which one?
Could you repeat the question, please?
So you see here a process in the image. So how is this process called? This one.
The transcription, there's a label.
Okay. Good. Yeah, that's transcription. Then we have some sort of RNA processing. Okay, so here's more tricky question. That's not labeled. How are these regions, these blue regions, how are they called?
The introns.
Yes, absolutely correct. So that's introns, and whatever is left is called
Exons.
Exons. Yes, correct. Good. So here we are. Here we see some cap on these mRNA, and here you see an poly A tail, and then it is exported into the cytoplasm, and here you see the process of translation and creating basically some protein. Okay, that's eukaryotic cell, but as I told you, I will be more focusing on prokaryotic cells, at least partially because we have no time to consider everything, and partially because all these processes, they are reasonably similar. Let's put it that way. So reasonably similar. Yes, there are differences, but these are not some principal differences. So if you understand one of the process for prokaryotes, you'll definitely understand that for eukaryotes. Here is one more illustration. That's a map for the chromosome of E. coli, and this map shows you the relative position of genes that are encoding many of the proteins important in DNA metabolism. So the number of genes known to be involved provides you a hint of the complexity of these processes. So many different genes. Even if you, well, maybe even some names may be already known to you. I'm not sure if you have considered that in from the point of view of bacteria, but if you have studied, for example, virology, you should definitely remember such things as GEC, POL, ENV, and other genes. So genes for envelope, for polymerase, so on and so forth. And here you see many genes for the bacteria. The numbers are called minutes, and they correspond to 40,000 base pairs along the DNA molecule. I'll be honest, I don't know why 40,000 was chosen, but that's what we have. Okay. Here is one interesting experiment to think about. So consider you are studying the duplication of the DNA. So we have the replication process, and we want to know how is it performed. So this is an experiment that basically tells you that the process of replication is performed semi-conservatively. So this means that you get two new copies, but each of these copies contains one of the old strains. So it may have happened, so before we knew anything about the DNA, that may have happened that we have some sort of old DNA, and there is some mysterious process that reads these double helix and creates you just brand new double helix. That was pretty reasonable assumption at that time. And there is another possibility that if you had your old DNA, you are creating two copies. Each of these copies contains one of the old strands and one newly synthesized strand of the DNA. And here is interesting experiment that shows you how that works. So here we have an original parent DNA molecule, and now we are performing, we are creating the first generation of the daughter molecules, but we use nucleotides that contain that contain nitrogen 15 instead of nitrogen 14. They can be later, they can be separated, and you can basically see what is the how many of these nitrogens you have in your DNA, and what is their relative weight or something like that. So here you see the heavy DNA, DNA 15, and here you see in the first generation you are creating hybrids, so DNA with DNAs 15 and 14. So if you isolate your DNA, you see only one band here. Can you tell me what would happen to this step if the other assumption was true? So if the process was not semi-conservative but fully conservative. So if we just created a new copy of DNA and the old one just remained as it was. Two strands. Yes, absolutely correct. We would have two strands in that case, but we have just one, which basically means that we have a semi-conservative process. And here we have the second generation, and here you see two strands, well, because you have two molecules that contain only new chains and two molecules that contain some old chains. Good. here is a process of replication, and here you see some micrographs. So this is a circular DNA, so of course you know that bacteria have circular DNAs, and here is the process of replication and how that looks like. So here you can see two replication forks, so replication fork 1 and replication fork 2, and they are moving in opposite directions, creating new copies of the DNA. And here is a schematic view on the bottom that basically shows you what happens to that strands of the DNA and which ones are old and which ones are newly synthesized. Okay. Here is this process enlarged. So what is the chemistry behind all that processes and what's happening in here? So here you see the strand that grows, and here you see the template strand. So you know that strands are directed. These directions are called 5' and 3'. This numbering comes from this sort of ribose ring. So here we have that, or desoxyribose ring. So here we have it. So here you have number 1, carbon number 2, carbon number 3, number 4, and number 5. Well, you have probably learned many different rules like direction of synthesis or reading or something else, and being honest, I find them all infinitely confusing because it seems pretty complicated to remember what is meant by each of that steps, so I'll be just as simple as possible. So the only thing I think you should remember to understand the process, that the chemistry behind that is the attack of the OH group on the phosphate. So on the 3' end you always have OH group, and this OH group attacks the phosphate, and thus the chain grows. So here you see how that looks like. So here I have oxygen here with the phosphorus, and oxygen attacks that phosphorus. We lose 2 phosphate groups and have one new nucleotide basically attached to the growing chain. So these 5' end gets attached, this part goes away, and we have new OH group that can be used for synthesis once again. And you do that all the time. You add something more and more, and you always have a free OH group that may attack something. So that's what we have. So that's why your 3' end always grows, so you always add new nucleotides to the 3' end, and of course if the template strand is anti-parallel, because strands are always anti-parallel, you read it in the opposite direction, so you are always getting closer to the 5' end of the template strand, and you always are adding nucleotides to the 3' end of the growing strand. Good. Here is a simplified version. So here you see the replication fork, and here you see how these strands are synthesized and the directions of these strands. So a new DNA strand, it's shown in light red, is always synthesized from 5' to 3' direction. So you always add to the 3' direction. The template is read in the opposite direction, so 3' to 5'. So you are always getting closer to the 5' because they are anti-parallel. And one more thing to notice, that the leading strand is synthesized continuously, while the other strand that we call lagging strand is synthesized in the fragments that we call Okazaki fragments. So the thing is that you can freely move in this direction, because you always have free 3' end, and you can always attach something new to that 3' end. While here we don't have that 3' end, so we have 5' end, and that's why we need to wait for some time and just start synthesizing from scratch. So that's why the lagging strand is synthesized in form of fragments. The Okazaki fragments are spliced together by DNA ligase. I guess I will show you the chemical formulae, how that works. And in bacteria, Okazaki fragments are typically from 1000 to 2000 nucleotides long. In eukaryotic cells, they are 150 to 200 nucleotides long. Here is one more picture that shows you that process. Here we have a DNA template. You cannot start synthesizing new DNA from nothing. So I hope you remember that to start that synthesis, we should add some primers. So at least some number of nucleotides should be added before we can set there a polymerase and start doing something. That is performed by the primase. Then we can use DNA polymerase, and this DNA polymerase starts synthesizing a new DNA. This primer is, in some sense, is left behind, and there are different polymerases that will replace this primer. So with different polymerase, this primer will be replaced, and then ligase will once again connect everything together. I guess we'll see that later. So here are the same images. Good. Elongation of the DNA chain. So here are a little bit more chemical details. So here is the catalytic mechanism. You can see it right here. So two magnesium ions are stabilizing and holding in place the new incoming nucleotide. So here it is. If we have a good base pairing and so on, the nucleophilic attack may be performed and we have a new bond created. These phosphates, they go away, and here we have a new nucleotide incorporated into the new growing strand. Here we have example of the DNA polymerase 1. It also requires a single unpaired strand to act as the template, and a primer strand to provide free hydroxyl group at the 3' end. And here is in this image you see the core of most DNA polymerases. It's shaped like a human hand. So this thing, well, I'm not sure. For me, that looks like more like a claw or something like that, but people say it looks like hand. So this part is called palm, this is thumb, and these are like fingers. And here's a cartoon representation of the polymerase structure that shows the insertion and post-insertion parts of the active site. So the insertion site is where the nucleotide addition occurs, and the post-insertion site is the site to which the newly formed base translocates. So we have here insertion site, new nucleotide is added, and then everything shifts. So here are some more examples. What happens if you have good and not very good base pairing? So a contribution of the base pair geometry to the fidelity of the DNA replication. So the standard AT and GC base pairs have very similar geometries, and an active site sized to fit one will generally accommodate the other. So they are well accommodated in that site. And the geometry of the incorrect base pairs can exclude them from the active site, as occurs on DNA polymerase. So here you see these examples of the incorrect base pairing, because everything is random. So all these molecules, they are moving pretty randomly, and you have no warranty that in your active site will not get some wrong nucleotide, so there's no one to gatekeep and check what is getting in there. So there should be certain mechanism that will provide at least at least some fidelity to the process. And here you see that they are not very well accommodated in that active site, so there is much more probability that the wrong nucleotide will just go away. But anyway, DNA polymerase inserts incorrect nucleotides time to time, and the statistics tells that it has one error for every 10 to the 4th or 10 to the 5th correct ones. So often these mistakes are due to the fact that the base is briefly in an unusual tautomeric form. So if you know some tautomery, how that looks like, so you can check different bases, and some of them have different tautomeric forms, and if they are in that sort of unusual form, they can fit where they shouldn't. But it's not everything. So we may have in DNA polymerases, we may have some error correction. So here is an example of error correction by 3' to 5' exonuclease activity of DNA polymerase 1. So here you see that exonuclease site and if polymerase mispairs, for example, C with T, so we have bad pair, the polymerase may reposition that mispairs the 3' terminus into the 3' to 5' exonuclease site. So here it is. And that exonuclease activity, it hydrolyzes the mispaired C. Here we see that, and we have once again we have free place to insert whatever fits there. So here you see this example of the error correction. So the DNA bound to the enzyme slides backward into the exonuclease site, and the enzyme corrects the mistake with its 3' to 5' exonuclease activity. When base selection and proofreading are combined, DNA polymerase leaves behind one net error for every 10 to the 6, 10 to the 8 bases. So that's really good additional accuracy that is provided by the exonuclease activity. Here is comparison of different DNA polymerases for E. coli, and here is a tricky question for you. Which row should I look to understand which one of these DNA polymerases is the main DNA polymerase? The one that synthesizes some large chunks of the DNA. The last one. Yes, the last one provides this information. So either the last one, the processivity, where we see that polymerase C or the DNA polymerase 3, it processes 500,000 nucleotides before it falls off, or the polymerization rate. You would expect from the main polymerase to work pretty quickly because it has a lot of work to do. Good. Neat translation. This is once again example of the DNA polymerase 1. It has, it seems that it has 3 domains that in these polymerase, and we have already considered the 3' to 5' exonuclease domain, but it also has 5' to 3' exonuclease domain. It's not shown here explicitly, but it does. And the interesting thing is that it may move along the strand synthesizing one strand and degrading whenever it has some strand in front of whenever you have some double strand in front of these polymerase. How do you think, why do we need that? You should probably know already, so so not so tricky question. Primer, yes, absolutely correct. So at some point we need to remove primers. You see, here is the interesting part. It's not because it cannot start with primer. It's because well, it's because other polymerase has left some primers behind, and you need some mechanism to remove that primers and substitute them with something more meaningful. So here you see we have some old strand, some polymerase, I don't know, polymerase 3 for example, has left this part behind. That was a primer. It synthesized some new strand, but this primer is left behind. Primer, that's basically what? RNA, yes, that's a small part of the RNA. And if you are trying to duplicate your DNA, you probably don't want some alien parts in there, so you don't want some RNA or whatever. So you need to remove everything that is not DNA and replace it with the DNA that is a perfect copy. And of course, here is one more tricky question for you. You already know that no DNA polymerase starts without primer. And how can it happen that this DNA polymerase replaces some primers? Where does it get its own primer, and who replaces that primer? There's one thing I want you to notice. So what's so special about the DNA of the bacteria and the prokaryotes? Yes, it's circular. It's circular. So if you are performing the duplication of the circular DNA, so I'm start replicating, I'm leaving some primer behind. So here is my sort of primer. Well, I'm oversimplifying, but you get the idea. I can go around, and now I use this newly synthesized DNA as the primer for the DNA polymerase 1, and it can move a little bit further and replace this sort of primer that was left behind with the DNA. So I displace it and I leave it as it is. Good. And what do eukaryotes do in this case? Eukaryotes don't have circular DNA, so how do we survive? Telomeres, yes, absolutely correct. So we have telomeres, and we may afford losing some part of that telomeres because there's nothing informative in that part of the DNA. In some cases, so-called telomerase may be active and may add something to the telomeres, but I'm not sure regarding the most recent information, because as far as I know that telomerase is incredibly active in case of cancer cells. So I'm not sure whether it does something during, let's say, normal operation. Maybe you do know, you have studied that more recently than I did. And in stem cells, maybe yes, I don't remember, I'll be honest with you. So I don't know everything, so yes. But there may be some problems with that, and you know that there is certain limit called Hayflick limit, that the number of the division that cell may survive, well, on average, of course, and that may be connected as well to the shortage of the telomeres. Good. Here is a nice picture for you. That's the DNA polymerase 3. We have already noticed that this is the main polymerase that does almost all the job by synthesizing the DNA. So it synthesizes the largest chunks of the DNA. So here is its architecture. You see here the clamp loader. Here you see sliding clamps or beta sliding clamps, and these are the polymerases. So you see this complex. It contains 3 polymerases. When we are synthesizing a new double strand, one polymerase is sitting on the leading strand and just synthesizes everything as it goes. The other one is synthesizing Okazaki fragment, and one is preparing to be loaded on the DNA lagging strand to synthesize another Okazaki fragment. And this clamp allows DNA polymerase to remain for a longer time, to be attached to the DNA strand, and as a result synthesize more of the DNA than if we didn't have this clamp. And here is a video for you.
DNA replication is carried out by a molecular machine called the replisome.
Immediately tricky question for you. Do you see this? Oh, bad color choice. Let me do you see this thing? What's that? Who can tell me? Helicase. Yes, absolutely correct. That's helicase or helicase. I'm not sure how to read that.
That pulls apart the double helix and makes an exact copy of each strand. In this visualization, we're going to examine the mechanism for copying DNA. The enzyme.
So which strand is leading, which is lagging? The one on the right or the one on the left? Can you recognize? No, the right is the leading strand. You see, the leading strand is synthesized continuously. See, it's synthesized continuously, so I have only one DNA polymerase here, and I'm just synthesizing, so that's the leading strand. And on the left, you see this sort of, where are the Okazaki? Absolutely good question. Let me maybe make one adjustment here. It's it's not very well seen, but you see this sort of DNA polymerase is loading, and here is, let me take another color. So here is the DNA strand that goes behind the thing, goes here, and here goes away. So these things are synthesizing the Okazaki fragments. So that's the lagging strand on the left. It's synthesized in chunks. Let me turn on the video. I guess maybe it will be seen a little bit better when you see that in the movement. So look at the left side. You see this part of the DNA, and that will be a new Okazaki fragment. So let me start that.
The mechanism for copying DNA. The enzyme DNA polymerase synthesizes DNA molecules, using the original molecule as a template for the production of a complementary strand. The genetic code in the template strand is paired to free-floating bases, matching cytosine to guanine and adenine to thymine. DNA polymerase creates an exact copy of your DNA code, making less.
Here is another shot, so maybe you will see in these frames, maybe you'll see that a little
than
bit
one
better.
mistake in a billion bases.
So here new polymerase attached, and here the synthesis should go on. Good. Maybe I have some more images, and we will look once more at that, at the Okazaki fragments and how that looks like. Good. Here is the model of the initiation of the DNA replication in E. coli. So here you see so-called locus ori, so the origin of replication. And here is the typical view of that locus ori and what we expect to see there. So here is a region DUE. That's the region where primary melting is performed. And here are certain conservative sequences that are found in the DNA. And here you see the direction with these arrows that tell you which direction is that sequence oriented, because you have two strands, so it may be along one strand or the other. So here you see the AT rich DUE region is denatured as a result of the strain imparted by the DNA A binding, and formation of the helical DNA A complex is facilitated by proteins HU, IHF, and FIS, and the detailed structural role of these proteins are not known, at least not known to me at this point. So maybe we have some more new articles that provide some insights. And here you see the DNA B helicase activity further unwinds the DNA in the preparation for priming and DNA synthesis. So here you see how that happens. So you have DUE region, you have that locus ori, you have DNA A binding, so that's origin binding. Here you see what happened. So HTH, IHF, they do something but here we get at the DUE region we have some melting of the DNA strand, and now it is prepared to load the DNA helicase. And DNA helicase will promote the further decoupling of the DNA strands. Even more, you will get not one but two DNA helicases, because you remember that we have two replication forks that go to different directions, so one to the left, one to the right. And we know that at least 10 different enzymes or proteins participate in the initiation phase of the replication. Here are at least some of them are listed and their tentative functions. What can they be? And in the case of DNA A, the ATP bound form is active and the ADP bound form is inactive. 8 DNA, 8 protein molecules, all in the ATP-bound state, assembled to form a helical complex encompassing the R and I sites in the OREC. So here you see that complex, the pre-priming complex. Here you see DNA A, here you see single-strand binding proteins, SSB, and here you see the helicases that are loaded. So interesting thing to notice that if we have certain proteins that are active with ATP bound and that are inactive with ADP bound, that means what? Recall our last lecture, we had there was one protein with pretty similar property. It was active with ATP and in some sense inactive with ADP. And why did we want that? No, don't remember. Okay, that was the G protein, and we wanted that because ATP can be spontaneously hydrolyzed to the ADP. So we have in some sense a timer installed into the protein that may turn that protein off. So if we don't want some protein to be permanently active, we need it to do some work and then stop, to not it anymore. So that's probably maybe a nice idea to create a protein that is active with the ATP bound. So after some time passes, ATP will be hydrolyzed to the ADP and your protein will just turn off. So in case of G proteins, we didn't want our cascade to be permanently active because we just wanted to pass signal that something came from the outside and we have noticed some changes in the environment, and then we want to stop that. And you remember that there are many medical conditions that involve some permanent activation of certain cascades. In this case, we have some proteins. We want to initiate the duplication of the DNA, but we don't want them to be permanently active. So we start it, and that's it. Good. Here are some. Here is a small table, so prokaryotes versus eukaryotes. So you see there are a little bit different DNA polymerases, but anyway, in principle, yes, they are different in some aspects, but the processes look to me reasonably similar, so we are not going into too many details. For copying a eukaryotic replicon, we need at least 2 DNA polymerases, an initiator polymerase called polymerase alpha that begins replication, but it's soon replaced with a more processive enzyme delta, and this process is called polymerase switching. I don't think we have something similar in prokaryotes, but that's that's it. Here is example of synthesis of the Okazaki fragments. So here you see our DNA helicase. Here you see the leading strand. So that's a strand that is just synthesized without interruption, more or less without interruption. So here you see the replication fork movement. Here is the DNA topoisomerase. Does everyone know what's that and why do we need topoisomerase? To uncoil strands. Yes. So if we have these double strands, we need somehow to solve the problem of them to be tied in some sort of knots. And I guess I have a nice video somewhere further in the slides. I will show you how that topoisomerase works. Well, at least one of the topoisomerases, because there are a few of them. Good, so here you see the replication fork, here you see SSB, so single strand binding proteins that are covering the DNA strand before we will perform some sort of synthesis on that strand. And here you see that so notice, if we consider the two template strands as lying side by side, lagging strand synthesis formally proceeds in the opposite direction to the synthesis of the leading strand. So we synthesize this direction while this polymerase moves this direction. So each primer is extended by the DNA polymerase 3. So here we have some primer. Here we have that DNA polymerase that extends the primer. It displaces the SSB proteins that were covering the single strand of the DNA. So you see, we should make some part of the DNA should be kept in a single-stranded form, and as you might guess, it's not really good for the DNA. So you may get some damage of the DNA strand if it's not base paired with the with another strand of the DNA. So to avoid certain problems, the cell has these sort of proteins that are immediately covering the single strand of the DNA, and then they are displaced by the polymerase as it moves along that strand. So here you see how the process proceeds. So synthesis continues until the fragment extends as far as the primer of the previously added Okazaki fragment, and a new primer is synthesized near the replication fork to begin the process again. And each DNA polymerase 3 whole enzyme has 3 sets of core subunits, so 1 or 2 Okazaki fragments can be synthesized simultaneously. So that's what we have seen. So this is the clamp loader. Here you see clamps, and these are DNA polymerases that are performing synthesis at the moment. So here you see 2 DNA polymerases synthesizing Okazaki fragments. Here is a new single-stranded DNA that will be primed, and DNA polymerase will synthesize something there. So here is the helicase, and this is the process as it goes. Good. Here we see the DNA polymerase clamp loader. So here you see that we have this part. So that's a clamp loader. This is a clamp. This is a clamp that will be loaded on the DNA strand. So here we have the continuous synthesis on the leading strand, as DNA is unbound by the DNA B polymerase, and here you see the lagging strand and the RNA primer of the previous Okazaki fragment. So here is primase. So primase creates a new primer. Here is primase attached to the single-stranded DNA. Here is synthesis of one of the Okazaki fragments, almost complete. So primase binds to the DNA B and synthesizes a new primer, and then dissociates. So here primase went away. A new beta clamp is loaded, so the clamp loader is now loading a new clamp. And here is shown that the synthesis of the Okazaki fragment was finished. Now what do we have? The polymerase may go away from here, and it can be reattached, for example, to the new clamp that is loaded. Or if we have 3 DNA polymerases attached to this complex, we can attach a new DNA polymerase, and this will just be free for some time, so that depends. And here we have beta clamp left behind. It will be later removed, and here you see that DNA polymerase starts synthesizing the strand starting from the primer. And here is process basically returned to the state as we had at the beginning. So So here you see this clamp loader a little bit closer. So the 5 subunits of the clamp loading or gamma complex are the delta and delta prime subunits, and the amino terminal domain of each of the 3 tau subunits. So if you consider that proteins a little bit closer, you will see many different subunits, and they all are named with Greek letters. So so whatever, the thing to remember, that complex binds to 3 molecules of ATP and to the dimeric beta clamp. This binding forces the beta clamp open at one of its 2 subunit interfaces. Hydrolysis of the bound ATP allows beta clamp to close again around the DNA. So we consume ATP because we need energy to set that clamp on the DNA. So that's the clamp loader. And good. What do we have here? So these are the final steps in the synthesis of lagging strand segments. So here you see the lagging strand. We have synthesized up to the primer. Then DNA polymerase 1 or RNase H1 may displace this primer. So you remember about that exonuclease activity, so it can displace this primer and synthesize everything up to this point. So now almost everything is okay, except that we need one more chemical bond to be created. So remaining nick will be sealed with DNA ligase, and I guess, yes, I have an image to show you. So here is the DNA ligase. It may be adenylated. So here you see the loading of the ribose, adenine and 1-phosphate group onto the DNA ligase. And here is what it is, what it should perform. So this picture is a little bit not up to scale. Here let me show you where it's not. So here you have base pairs. So these are base pairs, these are base pairs, and these are base pairs, and so on. So this gap that you see here, that's just a gap between neighboring base pairs. So there are no more base pairs in this gap, no more strange atoms, nothing like that. So basically this small distance between these 2 base pairs is the same as this large between these 2. Let me choose these colors. So this distance is the same as this distance. That's just drawn for the sake of illustration, so not to take the whole slide for one image or the whole page of the book where I have taken this from. So that's just one point to notice and not being confused. So we have all the base pairs, we have all in place. The only thing that we are lacking, we need somehow connect the 3' terminal OH with this 5' phosphate that we have here. And the problem is that we cannot do that just as it is. So you cannot attack with this OH, you cannot attack phosphorus and get a new bond. How do you think why? They are just neighboring one to one another, but there is no chemical bond created. So this OH does not attack phosphorus. How do you think, what can be the reason for that? You see, if I attack that color, sorry, let me choose this one. I have here a non-bonding electron pair. If I attack these phosphorus, so I'll be trying to create a new bond. Let me draw that like this. So I'm giving this electron pair to create a new bond. I will need something to go away from that phosphorus. And you see that the only living group I can get is getting these electrons to the oxygen and getting something really ridiculous like an atom of oxygen with with charge -2. Of course you cannot have that. So that's absolutely unstable and there is nothing you can do. But with the help of the DNA ligase, so here we have DNA ligase, it has accepted this thing and now after this DNA ligase has found a nick in the DNA, it replaces this group with this thing. So here you have ribose, adenine, and now you have 2 phosphates. And now if I attack with these non-bonding electron pairs, if I attack phosphorus, I can safely take these electrons to this oxygen because it's not just one oxygen, it's part of this phosphate group. So I'm not creating something ridiculous, I'm just creating regular phosphate group with 2 minus charges. And moreover, these sort of phosphate groups with minus charges, they are pretty stable because of what? Who will tell me the keyword? When such charged things are pretty stable, in most cases that's due to what? No, due to resonance. So remember about resonance and resonant forms. So I have drawn you many pictures previously, and I guess the one that I like the most, that's the image of, I don't know, some acid. So if you have something like this, you have OH group. If hydrogen leaves, we get O minus, but this minus charge is basically stabilized with the resonance stabilization. So you can have these electrons going down here and these electrons going up here. So which means you are getting something like this. So you get double bond here and minus here, and it is even that's often drawn as you have bond and something like partial bond between these 2 oxygens, so having some minus charge delocalized. That's the reason why you have the same OH in alcohols, but alcohols are not acidic. Acids are acidic. And that's because of the resonance. And here you have a very similar case, so these group will happily leave because you have that resonance stabilization. In case of bare oxygen, you don't have any resonance because there are no other atoms to delocalize your negative charge. So here it is. So the reaction goes, and you have this nick sealed. So here you see how that works. So we just make this part, let's say, reactive, and reaction goes good. Here is regarding the termination. So eventually the 2 replication forks of the circular E-coli chromosome meet at a terminus region containing multiple copies of 20 base pair sequence called TER. So here are different TER sequences in the in DNA. and their sequences function as binding sites for the protein TUS, so terminal utilization substance, and the two-star complex can arrest a replication fork from only one direction. Only one two-star complex functions per replication cycle, the complex first encountered by either replication fork. Given that opposing replication forks generally halt when they collide, their sequences would not seem to be essential, but they may prevent over-replication by one fork in the event that the other is delayed or halted by encounter with the DNA damage or some other obstacle. So these are these sort of traps for the replication forks. What else? Here is a nice video for you. So I have already told you about the topo isomerase. So let's look how that works. Here is a really
When we
nice
zoom
video.
in on the upper part of the topoisomerase, one of the two overlapping DNA segments enters the upper gate. It then binds to the middle gate. Now the second DNA segment enters the upper gate. The DNA in the middle gate is cut in two by the topoisomerase.
So you see what we want to do. We want to unknot somehow the DNA, and to unknot any knot, there is even a mathematical theorem about it, is that if you have some crossing, so here is one DNA double strand, here is another one, so they are overlapping like this. If you have the ability to make somehow one pass through another, so that you get this state instead of this state, you can basically unknot any knot. And that's what is this topoisomerase doing right now. So it takes one strand, it takes another strand, and it will break one of the strands, pull another strand, and seal that together again. So here, let's look how that works.
Two ATP molecules attach to the upper gate and the gate closes. One of the ATP molecules is broken apart into ADP and phosphate, releasing energy that may help keep the upper gate together during the next steps. The middle gate now opens and pulls the two halves of the broken DNA apart, creating a gap. Since the middle gate is
So now one of these strands of the DNA is broken, and you can safely move the other strand through that strand. So here is the moment when they are changing the order in which they overlap.
connected to the broken ends, the DNA remains attached to it. The second DNA segment now moves through this gap. The middle gate closes and the attached DNA ends are reconnected. The upper gate rotates, which may prevent the second segment from moving back through the break. The remaining ATP molecule breaks apart, and the lower gate opens, allowing the second DNA segment to leave. Then the lower gate closes, the upper gate opens, and the first segment of DNA is finally released.
Okay, so this video is a little bit longer, I guess. You can check it after the lecture, the whole video, so you will know a little bit more about the topoisomerases. Good. One more thing I want to show you about the DNA, that's DNA recombination. So that's an exchange of genetic material between DNA molecules that are generating new allele combinations. So increases genetic diversity in sexual reproducing organisms, enables proper chromosome segregation during meiosis, that contributes to the DNA repair and genome stability, and that even happens in prokaryotes. There may be this sort of DNA recombination. So even in that cases, you can have different processes like transduction, transformation, or conjugation in prokaryotes. So during some of these processes, you may have this sort of recombination and so on. But what's interesting, it's really it turns out that it's really beneficial to have this sort of process from the evolutionary point of view. so that you can move certain genes between the chromosomes, and that's pretty good from the evolutionary point of view. And how do you think, why? Why do we want to move genes between different chromosomes? So let's even forget about bacteria, I think, for a moment, well, about any organism that you feel comfortable with. So why do we may want to do that? Well, in the process of evolution, you get some evolutionary benefits if you can perform that recombination, as if compared to the case if you were not able to do that. So how evolution works? So you have a bunch of organisms. So they are living their life, doing something, whatever they do. You have some random mutations. Most of these mutations will do nothing. Some of the rest, well, most of the rest will kill organisms. But sometimes you will get some useful mutations that are pretty beneficial. What happens if in this bunch of organisms you get one organism with a really good mutation? What does it do? Good point. That's a good point. It has to do with some sort of recovery processes, but that's not exactly what I want. Yes, that organism in some sense becomes dominant. So it leaves more descendants, and after a few generations you have all organisms will have that beneficial mutation, because they have they have just dominated over the organisms that didn't have that mutation. So that's good. But what happens, think now, what happens if you have two organisms, each of them has some beneficial mutation, but these are different mutations? So in cases when we have just one organism with something pretty beneficial, we expect that after a few generations we will have all organisms containing that mutation. Well, you probably may expect that one of them will basically dominate over the other one, and the population will eventually lose one of the beneficial mutations. Well, do you see how that works? Yeah, so mutations are inherited by different organisms, but probably one of these mutations will be more beneficial than the other one, and so you expect that during the next generations, one of them will be probably lost. So it is just displaced. And the reason is that you have different organisms containing different beneficial mutations. And now you have DNA recombination. So what may happen? So with the help of DNA recombination, it may happen that these two beneficial mutations will be eventually collected in one organism, and they will be passed to the next generations now together. So now organisms with two beneficial mutations will displace the rest of the organisms, and at certain generation you eventually get organisms with two beneficial mutations. So you see how that improves the adaptability and the evolutionary adaptability of the organisms. So to get to beneficial mutations without this sort of recombination, you need what? You need to get one beneficial mutation, and when it's spread and these organisms have displaced all the rest, you need to wait for some time, because before one more beneficial mutation occurs. But in case of DNA recombination, it may happen that you eventually have in your population two organisms with two different beneficial mutations, and during some time, after a few generations, you may get some new organism that obtained both of these mutations. So you don't need to wait once again, because it will be a really long time. So mutations, it's really long. You need really long waiting before you get any beneficial mutation. So they are mostly, they do nothing, and in the cases they do something, they will mostly do something bad. So it's so you really want to retain that. So all the organisms that have mechanism of changing their DNA, of DNA recombination and so on, they have like evolutionary benefits from the ability to collect some useful mutations together and pass them as a whole bunch to the next generations. Okay. So that's the evolutionary point of view. I think so. Maybe that's something new I was able to tell you. And of course, you should already know that this sort of recombination, like homologous recombination, may be quite useful if you are repairing double-strand breaks and so on. And you need at least one recombination during meiosis. Maybe I'll show you that as well. Good, so here is recombinational DNA repair at the collapsed replication fork. Pretty may happen. So here we have DNA, and here we have a DNA nick. So DNA polymerase synthesizes, and at some point everything breaks down. So you have DNA nick, you have synthesized up to this point, and you cannot synthesize any further. So you just get some broken part of the DNA. So what can you do in this case? So here you see an example how that's handled. So you can get some 5' end processing. So some of these nucleotides are removed. Here you see the process of strand invasion. So you use some homologous DNA to basically fill the informational gaps in the broken DNA. So here is strand invasion. Here you see branch migration. So here is some bigger view. I guess I have some nicer pictures of that, including the holiday junction. But anyway, And here is the result of holiday junction resolution and ligation, and we can proceed with our DNA synthesis. So I guess I have some nicer pictures. We will get to that. Here is one more example. So this is a micrograph. Here you see bound proteins rec A. So in E. coli the DNA and processing is promoted by the rec BCD nuclease helicase, and the rec PCD enzyme binds to the linear DNA at the free broken end and moves inward along the double helix, unwinding and degrading the DNA in the reaction coupled to ATP hydrolysis. So the rec B and rec D subunits are helicase motors. So here you see how that looks like. So rec B, here you have rec D, a pin, and they are moving along the strand, and one of these strands is degraded. So the chi sequence is bound by rec C, preventing further degradation of the 3' end strand. So it's looking for certain sequences in the DNA, and when it finds this sequence, is something like this: GCT, GGT, GG. It binds tightly to the site on the rec C subunit, and from that point degradation of the strand with 3' terminus is greatly reduced, but the degradation of the 5' terminal strand is increased. So here you see how the degradation goes on, but we have one of the strands is blocked here, so it's not degraded, and we get just one strand of the DNA. So here you have degradation of both strands, so probably probably I have misspoken. So here is the degradation of the both strands. So these proteins are just chopping off whatever they meet. At some point they get to the chi site. This chi site is tightly bound, and the degradation of only the one strand is degraded from that point. So here you see the example in the previous slide. So we need to degrade one of the strands. So a little bit is chopped off from the end, and then one strand is degraded, and then all the rest procedures will be performed. So this process creates a single-stranded DNA with the 3' end, which is used during subsequent steps in the recombination. So here is a nice picture of the recombination, but I guess I have something more dynamic. Yeah, these are proteins that are basically pulling that DNA strands. So here you see this process, but I think I'll better show you this part. So here you see that's step 1, resection. So you have double strand break, and you want to repair that break somehow. So you are degrading ends, so a little bit is chopped off here as well, but anyway, then one of the strands is degraded much more than the other one. The second thing, if we have some homologous DNA, this part of the strand, so single-stranded part, may look for its homolog in that DNA, and at some point it may find it. So here it finds its homolog in the DNA. If that homolog was found, they may bind. So just base pair one with another. So the strand has basically invaded. So that's strand invasion. Now the other strand is displaced and may bind somewhere else. And now we can synthesize some DNA. So here you see that DNA synthesis as it is performed. Now you have so-called branch migration. So you see how these branches, these are holiday junctions, and they migrate along the DNA. So here you see that in a little bit more details. So here is holiday junction, here is, and that's how it looks like. So these DNA strands are connected and you can view that in from different perspectives. If you think about that in 3D, so you may see this picture or this picture, and that's basically what is performed with these sort of proteins. So after strand invasion, branch migration is promoted by the complex called RUV-AB, and once a holiday intermediate has been created, it can be cleaved by a specialized nuclease called RUV-C that nicks the sealed DNA ligase, and the viable replication fork structure is thus reconstructed. So in case of your replication fork has collapsed in bacteria, you can fix everything with these complicated mechanisms. So here you see all these proteins, how they are making the branch to migrate, and here you see that junction may be cleaved and you get two different DNAs. And here is an example of what can be a result of that sort of cleavage. So probably the simplest results that's more characteristic of the eukaryotes and just applies to small portions of the DNA. You may have a dissolution. So they just reattach to their own DNA strands. In other cases, you may have so-called patch products or non-crossovers. So here are non-crossovers. So here you see some newly synthesized parts. Here we break this DNA, and that's what we get. And here are so-called crossover DNAs. So these are DNAs with the crossover. You might guess they are crossover and non-crossover if you look where do you look? If you look at this part and this part. So you see we started with double strands, with gray double strands, and we ended with gray double strands. So here is some sort of mess, but that's, we were fixing some errors, so of course something strange is happening here. Here we started with blue strands, and we are finishing with blue strands. In this image you can see the same thing. If you wait until the animation goes down, you will see that gray and gray, or blue and blue, so whatever. In case of crossover products, you see here are gray strands, but you will end up here with blue strands. So this is just some small portion, but if you have a large chromosome, so there is a lot of information written here, you basically switch half or even more of that chromosome for something different. So do you see how that is working? Omnious silence. Okay, I hope that means yes. Or you can look a little bit later on these images and I don't know, so think of them more deeply. Good. Here is just an illustrative slide. So here is mitosis and meiosis. And the thing is that during meiosis, you need to have this sort of connection between homologous chromosomes, because we are duplicating the DNA material, and then we are performing two divisions during the meiosis. And during the first division, we need to somehow understand where are that sister chromatids. And that is with the help of all that processes that I have shown you. So this sort of this sort of connection between chromosomes. Good. So here you see that even in the image, so here you see this result of the homologous recombination. So you need homologous recombination to perform meiosis without certain problems, because you need to distinguish between that chromatids, and only the homologous recombination gives you a hint which chromosome is sister chromatid to which another chromosome. Good. Here are two videos for you. So since we are scarce on time, I guess I'll leave this one for you. That's DNA packaging in the eukaryotes. Maybe you'll know. Maybe you'll see some nice pictures. Here is a little bit more about so-called gel electrophoresis. I guess I'll show you just. Maybe I'll take a few more minutes just to tell you something about methods, some experimental methods. So let's oh. Okay, I'm not sure what's going on with these slides. It's like replication fork collapsed. Good. And then tomorrow we will start with RNA synthesis and all the rest. Good. So just so DNA electrophoresis. Here I have found a nice slide for you that shows what you can use the electrophoresis for. Basically, there are different approaches that allow you to work with DNA or RNA or protein, so on and so forth, and they are all called blots. So you see southern blot, northern blot, and so on. Southern, that comes from the surname Southern. The rest, I guess, were called just because Southern sounded cool, so they proposed why not Northern or Western. So here is here is an example of the electrophoresis. It is performed in the agarose gel. So this agarose gel, you are it's submerged in a tank filled with salt solution that conducts electricity. So we will be using electric field to separate some parts of the molecules or some molecules. And using a pipette, you are loading, in this case, DNA samples into slots that are made in the agarose gel. The DNA samples are colorless, but you can add some sort of tracking dye. The main idea is that since you don't see what is happening in there, it may happen that you will be running your gel for too long, and your DNA molecules just will escape from the agarose gel. So you definitely don't want that. So you will probably add some dye to have at least some idea of where can they be. You cannot be ever certain, but at least something. So here you see the pipette, and you are adding that sort of samples. So the phosphate group in the DNA backbone, they carry negatively charged oxygens that we have seen in many our formulae. That gives the DNA molecule overall negative charge. So you might expect that negative charges would like to move to something positive. So if in an electric current the negatively charged DNA moves toward the positive pole of the electrophoresis chamber. Depending on the size of that chunk of the DNA, in some cases on its form, so there are different methods to perform that sort of electrophoresis. So you may add some more chemicals that in some cases unbind the DNA, or something like that, but anyway, you will expect that smaller molecules will move much faster than some larger molecules. So if you have different chunks of your DNA, you will expect the smaller segments to migrate faster and further, and during the time of electrophoresis. And then if you look at the result, you will see which sizes of the DNA molecules you have. So here is an example. Here they are migrating through the agarose gel. So now ethylium bromide is added. It binds to the DNA double helix and glows when illuminated with the ultraviolet light. So you can see where are these pieces of the DNA. So if you look at the image that you have, you will get something like this. So here you see one lane, it uses marker, so that just to give you some approximate idea about the sizes of DNA and where they have migrated. So as a marker you use certain chunks, but you definitely know what are the sizes of that chunks. So when you see them on the lane, you can guess that this is the distance that the chunk of size, I don't know, 100 nucleotides should have migrated. And here you see your experimental samples, and by comparing them to the marker you might guess what is their size. So what are they? So the size of any DNA fragment can be determined by this sort of mapping. I'm showing this to you because I expect you to solve a few simple problems that involve, well, knowing something about the gel electrophoresis. And here is one more interesting assay. The thing is that we are always trying to check our hypothesis, and if you have the protein and you expect it to somehow, I don't know, unwind the DNA, or you have some number of proteins that you expect to unwind that DNA, here is a sort of assay that allows you to check that hypothesis. So first of all, you can label one strand of the DNA probe. You bind the proteins to the DNA and you treat it with the single-stranded DNA cleaving reagent. So maybe some sort of endonuclease or potassium permanganate. So that depends. These sort of molecules, they are pretty small, so they can get into all the nooks and crannies. So the fact that some proteins are bound to the DNA, it's not bad for this molecule. They will still get into there and do their work. And their work is to oxidize the unpaired thymidines, so T's, and these can be then chemically cleaved. So if you treat them with a very strong base, let's say piperidine, and that will basically cut that sides of the DNA. So you see what's happening. If you have these T's in pairs, so they are base paired to the to another strand, this permanganate will not get to them, and they will be not oxidized. If they are not base paired, so if they exist at the single strand, they will be oxidized. So then if you treat that after that, if you treat them with strong base, you get cuts in exactly in that places where you had these sort of T's. So if you perform everything that, if you get rid of the protein, you concentrate the DNA, and you separate DNA on a denaturing acrylamide gel. By denaturing, I mean that these strands will be unwound, so you unwind the DNA and you separate them on the agarose gel. You might guess that you will get lots of different chunks of the DNA that which sizes depend on where your DNA was cut. And in case if the proteins that you were suspecting of having some unwinding activity do not have this activity, you will see what? Who can predict? So here you see this label. So here is the label. You get the same DNA. You get your proteins, but your proteins do not perform any unwinding. So what happens? Will potassium permanganate be able to oxidize something? I'll just go step by step. Probably not. Yeah, absolutely correct. It will probably be not able to oxidize something because you will probably have no unpaired teeth in that chunk of the DNA. So if it's not able to oxidize them, then you treat with piperidine, and it does not cut at that sites. So there are no cuts in the DNA strand. So when I separate everything on the denaturing acrylamide gel, I will see what? So DNA is still there, so and label is still there, so I should be able to see that label somewhere. But I will see only one band, so only one band, and that's it. So let me choose some better color. And that band should correspond to the large chunk from the beginning to the end. Because I had no unwinding activity, this treatment with potassium permanganate did not do any oxidation, and thus cleavage were not successful, so no chunks were cleaved. If it has some unwinding activity, I have some cleavage sites, and I should be able to see something like this, so some small chunk. A different DNA molecule will be not so lucky to get cut here, but it will be cut somewhere here, so I'll get some larger chunk somewhere here, for example. Some other will be cut somewhere here, so I get some even larger chunk, and so on and so forth. But in case there is no activity, I get just one chunk, so just one band in the lane where I am doing something with this DNA. Good. Okay, I guess I guess with that being said, we can complete today's lecture and continue tomorrow. If you have any questions or comments so far, please fire away. Okay, no questions, no comments. I guess everyone's tired, so I understand. The semester is going to the end, so let's wrap up. So have a nice evening, and meet you tomorrow.